Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Apicoplast. 12 languages. An apicoplast is a derived non-photosynthetic plastide found in most apicomplexa, including Toxoplasma gondii, and Plasmodium falciparum and other Plasmodium spp. Parasites causing malaria, but not in others such as Cryptosporidium. It originated from algae through secondary endosymbiosis. There is debate as to whether this was a green or red alga. The apicoplast is surrounded by 4 membranes within the outermost part of the endomembrane system. 1. The apicoplast hosts important metabolic pathways like fatty acid synthesis, isoprenoid precursor synthesis and parts of the heme biosynthetic pathway. 2. Significance, edit. Apicoplasts are a relict, non-photosynthetic plastide found in most protozoan parasites belonging to the phylum Apicomplexa. 3. 4. Among the most infamous Apicomplexan parasites is Plasmodium falciparum, a causative agent of severe malaria. Because apicoplasts are vital to parasite survival, they provide an enticing target for antimalarial drugs. 5. Specifically, apicoplasts' plant-like properties provide a target for herbicidal drugs. 4. And, with the emergence of malarial strains resistant to current treatments, it is paramount that novel therapies, like herbicides, are explored and understood. 5. Furthermore, herbicides may be able to specifically target the parasite's plant-like apicoplast without any noticeable effect on the mammalian host's cells. Citation needed. Evolutionary origin, edit. Evidence suggests that the apicoplast is a product of secondary endosymbiosis, 6, and that the apicoplast may be homologous to the secondary plastide of the closely related dinoflagellate algae. An ancient cyanobacterium was first engulfed by a eukaryotic cell but was not digested. The bacterium escaped being digested because it formed a symbiotic relationship with the host eukaryotic cell. Both the eukaryote and the bacterium mutually benefited from their novel shared existence. 7. The result of the primary endosymbiosis was a photosynthetic eukaryotic alga. A descendant of this eukaryotic alga was then itself engulfed by a heterotrophic eukaryote with which it formed its own symbiotic relationship and was preserved as a plastide. 8. The apicoplast evolved in its new role to preserve only those functions and genes necessary to beneficially contribute to the Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF host organelle relationship. The ancestral genome of more than 150 kb was reduced through deletions and rearrangements to its present 35 kb size. 4. During the reorganization of the plastide, the apicoplast lost its ability to photosynthesize. 8. These losses of function are hypothesized to have occurred at an early evolutionary stage in order to have allowed sufficient time for the complete degradation of acknowledged photosynthetic relicts. 4. And the disappearance of a nucleomorph. 8. Architecture and distribution, edit. Most apicomplexa contain a single ovoid-shaped apicoplast that is found at the anterior of the invading parasitic cell. 4. The apicoplast is situated in close proximity to the cell's nucleus and often closely associated with a mitochondrion. The small plastide, only 0.151.5 m in diameter, 4, is surrounded by 4 membranes. 8. The 2 inner membranes are derived from the algal plastide. Membranes. 4. The next membrane out is called the periplastide membrane and is derived from the algal plasma membrane. Finally, the outermost membrane belongs to the host endomembrane system. 9. Within the apicoplast's stroma is a 35 kb long circular DNA strand that codes for approximately 30 proteins, tRNAs and some RNAs. 8. Particles suspected to be bacterial ribosomes are present. 5. The plastide, at least in the Plasmodium species, also contains tubular whorls of membrane that bear a striking resemblance to the thylakoids, 4, of their chloroplast relatives. 8. The import of proteins into the apicoplast through the 4 membranes occurs through translocation. Complexes that originate from the algal plastide, for example, 10, or from a duplication of the endoplasmic reticulum-associated protein degradation, for example, 11. Function, edit. The apicoplast is a vital organelle to the parasite's survival. 4. Tetracycline, an antibiotic also used to combat malaria infections, is thought to function by targeting the apicoplast. 12. It hosts 4 main metabolic pathways. Fatty acid synthesis, edit. The destruction of the apicoplast does not immediately kill the parasite but instead prevents it from invading new host cells. This observation suggests that the apicoplast may be involved in lipid metabolism. If unable to synthesize sufficient fatty acids, the parasite is unable to form the parasitophorous vacuole, PV, that is imperative to a successful invasion of host cells. This conclusion is supported by the discovery of type 2 fatty acid synthase, FAS, machinery in the apicoplast. 5. Isoprenoid synthesis, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The apicoplast is also thought to have a role in isoprenoid synthesis, which are prosthetic groups on many enzymes and also act as precursors to ubiquinons, involved in electron transport, and dolicals, involved in glycoprotein formation. 1. The apicoplast contains the 2,C-methyl-d-erythritol 4-phosphate, MEP, /1-deoxydesyllulose 5-phosphate, DOXP, pathway for isoprenoid precursor synthesis and is the sole site for such synthesis in the plasmodium cell. 1. Hem synthesis, edit. The apicoplast has also been implicated with hem synthesis, 5, and amino acid synthesis. It is also suggested to have a role in cell development. These functions, however, are merely postulations and are not yet conclusively supported by experimentation. 4. Iron-sulfur cluster synthesis, edit. Various iron-sulfur cluster biosynthetic enzymes including SUF-B or ORF470 have been identified in the apicoplast genome. 1. References, edit. 1 ABCD Lim L, McFadden G.I., March 2010. The Evolution, Metabolism and Functions of the Apicoplast. Philosophical Transactions of the Royal Society of London. Series B, Biological Sciences. 365, 1541 74963 do. 10.1098/RSTB.2009.0273 PMC 2817234. PMID 201243442. 2 Sheena L., Vadia AB, McFadden G.I., August 2013. The metabolic roles of the endosymbiotic organelles of Toxoplasma and Plasmodium spp. Current opinion in microbiology. 16, 44528. Do. 10.1016/j.mib.2013.07.003. PMC 3767399. PMID 239278994. 3 Reese JB, Yori LA, Kane ML, Wasserman Sat, Mineski PV, Jackson RB, 2011. Campbell Biology. 9th ed. Boston, Benjamin Cummings. ISBN 978-0-321-55823-7. OCLC 1008837408. 4 ABCDEFGHIJ Mary Chally, Cisprindilo MF, May 2001. The Apicoplast, a new member of the plastide family. Trends in Plant Science. 6, 5205. Bibcode, 2001 TPS, 6200M. Do. 10.1016/1360-1385, 01, 01921-5. PMID 11335172. 5 ABCDE Ralph Sat, D. Ombrin MC, McFadden G.I., June 2001. The Apicoplast as an antimalarial drug. Target. Drug resistance updates. 4, 314551 do. 10.1054/drup.2001.0205. PMID 11768328. 6 Ralph Sat, Foth BJ, Hall N, McFadden G.I., December 2004. Evolutionary pressures on Apicoplast. Transit peptids. Molecular biology and evolution. 21, 12 2183-94 do. 10.1093/Molbev/MSH233. PMID 15317876. 7 Alberts B, Brady, Hopkin K, 2004. The Eukaryotic Cell. Essential Cell Biology, 2nd ed. New York, London, Garland Science, Taylor and Francis Group. ISBN 978-0-8153-3480-4. OCLC 8952549551. 8 ABCDEF Kimball J.W., the 16th of December 2017. Endosymbiosis and the Origin of Eukaryotes. Kimball's Biology Pages. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 9 Sheena L., Strippen B., February 2013. Protein sorting in complex plastids. Biochimica IT. Biophysica Acta, BBA, Molecular Cell Research. 1833, 23529. Do. 10.1016/j.bamca.2012.05.030. PMC 3494742. PMID 226836761. 10 Sheena L., Fellows J.D., Ofsirikova J., Brooks C.F., Agrawal S., Holmes Z.C., Byatz I., Flinner N., Heine S. Myris O. Patiborski J.M., Strippen B., December 2015. Toxoplasma gondii TARC 75 functions in import of stromal but not peripheral apicoplast proteins, PDF. Traffic 16, 12 125469. Do 10.1111/tra.12333. PMID 263811927. 11 Agrawal S, Van Doren GG, BTWL, Strippen B, November 2009. Genetic evidence that an endosymbiont-derived endoplasmic reticulum-associated protein degradation, ERAD, system functions in import of apicoplast proteins. The Journal of Biological Chemistry. 284, 4833688391. Do 10.1074/jbc.m109.044024. PMC 2785210. PMID 198086883. 12 Dal L, Shock JL, Sheen IBR, Gut J, Derisi JL, Rosenthal PJ, September 2006. Tetracyclines specifically target the apicoplast of the malaria parasite Plasmodium falciparum. Antimicrobial agents and chemotherapy. 50, 9 3124-31 Do 10.1028/AAC.00394-06. PMC 1563505. PMID 169401111. Categories: Malaria organelles, endosymbiotic events. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.